So for today, we will have our discussion about the enzymology. And this will be the first topic for the entire SEM, right? So ito yung unang topic natin for this semester, enzymology. And actually, mahaba to. That's why we have allotted 2 meetings for this. So for today, we will have the introduction and some of the specific enzymes. And then by next week, the remaining specific enzymes. Okay? So enzymology here is a branch of biochemistry which studies how the enzymes work. How the enzymes work through the relationship between the structure and the function, and kung paano sila nagfo-fold into their native state. Okay? So, ang focus natin dito definitely are the enzymes. So, anyone from the class, kung merong idea, ano ang enzymes? What are enzymes? By the way, if you are cold, you can unmute yourself or you can put your answer in the chat box. Yes, Gonzalez, Vincent.
Ma'am, they can be a catalyst po in a biochemical
Okay.
reaction.
Yes, very good. Thank you for that. Now, they can be a catalyst for the biochemical reactions. And some of your classmates also answered here, proteins, biological catalyst. So, yan yung mga keywords natin, right? So, you have here, they are also proteins, protein that has in the chemical reaction. Christian here answered, enzymes speed up the biochemical reactions. So, very good. Most of the answers here are correct. May ibang medyo naiba lang, but the rest of the students answered correctly. No. So enzymes are proteins. They are proteins produced by living cells which has in the biochemical reactions. No, biochemical reactions. So they are biological catalysts. So they are mostly found in all body tissues. So mamaya, we will be discussing the specific enzymes that can be found in a certain tissue, no? And they are appearing in what sample? Sa tingin ninyo? Sige, let's change the question. If we want to measure this, what type of sample are we going to collect? Or are we going to test? So very good, Syra, no? Blood samples. Specifically, your serum sample. Because the enzymes appears in the serum following cellular injury. So it could be a sign that there is a damage in an organ or meron tayong disorder or may disease or certain disease or various disorder yung ating patient. So nagkaroon ng organ damage. And also, pwede din yung ating plasma. We can also use the plasma as the sample, lalo na yung mga enzymes na involved sa coagulation. Okay? So again, enzymes are specific biologic proteins that catalyze biochemical reactions. And ano pa ang dapat tandaan sa enzymes? The enzymes are measured in terms of their activity. Okay? So, minimeasure natin to in the laboratory based on their activity and not on their absolute values or their concentration. Okay? Ang minimeasure natin dito or what we are looking for in the laboratory is their activity. So, bakit yung activity nila? Because again, they are catalysts. So we instead measure their activity kesa doon sa absolute values or concentrations. Okay? So we have now here our unit expected outcomes. At the end of this topic, you are expected to be able to define the enzymes and their classifications. So mamaya, discuss natin yan. Discuss different factors affecting the rate of an enzymatic reactions because we have a lot of factors that could affect. And then discuss why the measurement of serum enzyme concentration is clinically useful. Okay, clinically useful. Actually, na-mention ka na, no? They are correlated to various disorders. So, their increase in activity could be correlated to certain disorders. And discuss which enzymes are useful in the diagnosis of various disorders. So, papasok yan dun sa mga specific enzymes, no? We will be discussing a lot of enzymes. So, first, we will have here the general properties of enzymes. General properties of enzymes. Again, an enzyme catalyzes many specific physiologic reactions. No? Ang sabi nga ni Christian kanina, they accelerate the rates of the chemical reaction in the body. And ano sabi niya? Without being consumed in the process. So they are not, or they do not become chemically altered in these reactions. So for the acceleration, kasi nga nag-accelerate, right? For the acceleration, it may occur in a solution, and the process is called homogeneous catalysis. Homogeneous catalysis. Now, if the catalysis or the acceleration occurs in an insoluble surface, the process is now termed as your heterogeneous catalysis. So enzymes, they are biological catalysts, no? So for the catalysis, kasi proteins to, right? For the catalysis, ang ini-exhibit nila is homogeneous catalysis. Homogeneous catalysis. However, there are some enzymes na embedded sila in the membrane structures which are considered to be insoluble. So meron din tayong mga enzymes na heterogeneous catalysts. Heterogeneous catalysts. Okay? So take note of that, no? So next now, again, we have here the properties, no? So enzyme is considered a protein. So as a protein, each enzyme can have a structure, right? You still remember our discussion of proteins in the clinical chemistry 1? Okay, yes. So we have their different protein structures. So babalikan lang natin, no? You have your primary, you have your secondary, tertiary, and some have quaternary. So as you can see here, first you have your primary protein structure. Primary protein structure here contains a specific amino acid sequence. Ayan siya, oh. Sequence of chain of amino acids. So, simple lang siya. Chain lang siya. Nandyan, no? So, you have here the amino acids. Now, if these chains will twist or will undergo twisting, it will result to your secondary protein structure. So, you have here your secondary protein structure. So, nag-undergo sila ng twisting and they are being linked by hydrogen bonds. Okay? And if this twisting will fold, nagkaroon na ng folds, it will result to structural cavities at ito na yung tinatawag nating tertiary protein structure. Okay? Tertiary protein structure. Now, if the enzyme contains more than one polypeptide unit, the quaternary structure results. Quaternary structure results. So we're already done with this in your clinical chemistry 1, right? So you have your primary, secondary, tertiary, and quaternary structures. Okay, so what else for the general properties of enzyme? You have here the terms active site and the allosteric site. So take note that each enzyme contains an active site and an allosteric site. And what do we mean by active site? Anyone? If you want to answer, you can also raise your hand, no? Para hindi ako magtawag, or kung wala, sige, tatawag tayo. Yes, Maureen Advincula.
Ano po, yung enzyme po na nagbabind sa protein or other substances po sa sa reaction.
Enzyme na nagbabind sa protein for the reaction to occur. Is that what you mean?
Yes.
So parang ganun, no? So medyo malapit na doon, no? Ito-tweak natin. Active site, oo, enzyme siya. But to be specific, it is a part of enzyme. Okay, it is a part of the enzyme. And saan siya makikita? Nasa surface region siya ng enzyme natin. Okay, this is the active site is the surface region, no? Nasa surface region siya. And this is where the binding and the catalysis occur. So very good, no? I have here a lot of answers in the chat box. So very good, Christian, Adrian, Angela, Sophia, Charm, Benedict, Angeli, and Christine George. So dito po sa active site, nagbabind yung ating substrate. This is where the substrate binds. And the shape of this active site here is a vital determinant in the recognition and specificity process. Importante siya. Okay? Because this is also where the substrate binds. So, importante yung shape ng ating active site. Okay? So, how about the substrate? What is a substrate? Cachero? Daniel? What is a substrate? Okay, very good. Where the enzyme acts? Kristen also answered here, the one that reacts with the enzyme. So Adrian answered can be protein or non-protein molecule. So very good. When we say substrate, it is a molecule for which a particular enzyme catalyzes a reaction. And is this a part of enzyme? No, this is not part of an enzyme. Okay? Siya lang yung nagbabind. So when they combine together, pag nagbind na sila, they will form your ES complex. What do we mean by ES complex? That is your enzyme substrate complex. And once na nagkaroon nito, this is where the reaction will occur and it will yield products. No, it will yield products. Sometimes the substrate will be degraded. So, nade-degrade sila. And then, this reaction here will result to formation of products. Okay? So, what are the other part? Or what is the other part? The other one is your allosteric site. So, this is a cavity other than the active site. So, nandito siya sa iba. And this is not where the substrate will bind. Hindi po nagbabind dyan. So, may certain amino acids present kasi sa active site wherein nagkakaroon ng affinity si substrate. So, dito siya na-attract, no, sa active site. Okay? So, those are the properties of enzymes. And again, enzymes are released when there is a damage in an organ or cells, impaired removal of enzyme from the plasma, increased permeability of cell membrane, and increased production of cells. So, kaya natin sila minimeasure in the laboratory. Because if there is an abnormal or large amount of enzymes in the serum samples, pwede silang maging clinical evidence of an organ damage. So, pwede natin silang gamitin. Okay? They have clinical significance. So, next now, we have this term, isoenzymes. Any idea about isoenzymes? Aaron, James. Malikdim. Any idea about isoenzymes? Yes, do you have an idea? Not sure. Are what do you mean? Are you not sure with your idea that you have in mind? Share mo lang kung ano yung idea mo about this isoenzymes here. Okay? So sige, pagka sinabing isoenzymes, these are enzymes. Ayan na with same catalytic function but different forms. So madami tayong sagot dito, no? Enzymes with the same structure, actually they do have different forms, no? Enzymes with same reaction but different forms, this is correct, Lara. Catalyze the same reaction, so Lara, Gemina, tama. Charm different amino acid sequence but their functions are the same. So very good. No, they have the same catalytic function but they have different forms. So kumbaga, may specific enzyme tayo and that enzyme may exist in different forms and that different forms are called isoenzymes, no? So, meron tayong multiple isoenzymes. Depende yun sa type of enzyme na yun. So, we will be discussing specific enzymes later on. Malalaman nyo kung sino yung mga enzymes na may isoenzymes, no? So, bakit nagkakaroon nito? These different forms here. These different forms occur because of differences in the amino acid sequences of enzymes. So, tama kanina, yung nasabi ni Charm, no, different amino acid sequences sa enzymes natin. But despite the differences in the sequence, the enzymes fold to the same three-dimensional structure and they frequently exhibit similar affinities for the substrate. And pareho din yung catalytic rates nila with the substrate, no? So, ang different forms natin, it can be differentiated from each other based on certain physical properties. So, you can have the electrophoretic mobility, the solubility, the resistance to an activation. Okay? So, one example of an enzyme with multiple isoenzymes and widely recognized na mga isoenzymes are usually yung mga composed ng varying combinations of subunits. So one of the most widely recognized is your LDH or your lactate dehydrogenase. Your lactate dehydrogenase here has 5 major isoenzymes and they are called your LDH1, LDH2, LDH3, LDH4, and LDH5. So your LDH1 is a combination, it is made up of 4 H subunits because your LDH here is made up of 2 subunits, your H subunits and your M subunits. Your H subunits and your M subunits. And your LDH1 here is made up of 4 H subunits. H, 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 H. Tetrameric molecule kasi ang LDH natin. Okay? Tetrameric molecule. So, 4. So, you have your LDH2. It is made up of 3 H subunits and 1 M subunit. For the LDH3, we have 2 H subunits and 2 M subunits. M. LDH4, 3 M subunits and 1 H subunit. LDH5, we have 4 M subunits. So you can see here that for each isoenzyme, iba-iba yung forms nila. So magkakaiba yung form ng mga to, but they have the same catalytic function. Pare-pareho sila. So these are the isoenzymes of your LDH or your lactate dehydrogenase. Now, paano po sila differentiate from each other? So itong mga to, they are being differentiated from each other based on electrophoretic mobility. So kung paano sila mag-migrate towards the anode. So anong ginamit nating method dito? Electrophoresis, right? Electrophoresis. So for the electrophoresis, we will be checking the migration no towards the anode. So sa electrophoresis or electrophoretic mobility, pagka itong ginamit nyo, ang designation natin is 1, 2, 3, 4, and so on, no? Kung ilan man yung isoenzymes na yan. So kung sino yung pinakamabilis mag-migrate, yun yung i-assign na isoenzyme 1. During electrophoresis. And kung sino yung pinakamabagal, kung ilan man tong isoenzymes na to, no? Kung sino yung pinakamabagal mag-migrate, the slowest one will be assigned the lowest number or the last number kung ilan man sila, no? So for example, here in your LDH, 5 silang isoenzymes. Si HHH, si HHHH, si HHHM, HHMM, HMMM, and MMMM, right? So during electrophoresis, chinek kung sino yung mabilis mag-migrate. So pinakamabilis mag-migrate ay si H4 or HHHH. So siya yung inassign na isoenzyme 1. Okay? So ganun sila differentiate. And yung pinakamabagal na mag-migrate, the slowest, is your